Yeah, good afternoon, everyone. And yeah, we're looking for unmuted. Correct. Okay. Good afternoon, everyone, and welcome to this week's quantitative seminar. I'd like to begin first with a land acknowledgement. The University of Washington acknowledges the Coast Salish peoples of this land, the land which touches the shared waters of all tribes and bands within Suquamish, Tulalip, and Muckleshoot nations. I'd like to introduce today's speaker, Dr. Brice Semmens. Brice is a professor at Scripps Institution of Oceanography and former director of the California Cooperative Oceanic Fisheries Investigations CAL-COFI program. Prior to his time at Scripps, Brice was an NRC research postdoc at NWFSC Eli Holmes and a statistical contractor at the Senate. He received his PhD from UW with Dr. Jennifer Rusing, his master's at UC Santa Barbara, and his undergraduate at Texas A&M University. And remind me again, did you say we did like questions during or after the presentation?
Whatever, let's do it during.
Let's do it during the presentation.
Sounds like that's more common. So.
Sounds great. So thank you. And without further ado, please welcome Dr. Brice Semmens.
Hey, you can hide those meeting ones that are across the slides there.
Yeah, you do that.
Just click on the three buttons on the right.
Hide video panel. Hide flooding media control. Good. Thanks, Mark.
Okay. Hi, everybody. This is a different quant seminar than it used to be. This used to be fall. This is post-COVID, I guess, right? I'm targeting about 45 minutes. I apologize. I kind of kludged this together at the last minute. I was dealing with some house stuff. So hopefully this will go largely according to plan, and you'll get something out of it. So, okay, let's start with begin with citizen science. So, how many people are birders here? Okay, how many people have done eBird? Okay, so eBird. Is for those of you who don't know or haven't done it, is a international program run out of the Cornell Bird Lab. And it allows birders to go and record all of the different species that they see, as well as the number of individuals of each species, and put that into a centralized database. That program is actually crafted on top of this program, which is the Reef Environmental Education Foundation's Reef Fish Survey project. This project was actually precedes the eBird program by about 10 years. Now, eBird these days completely swamps the data production of diving-based citizen science. Not surprisingly, there's a whole bunch of birders out there that have nothing better to do than go out with their $3,000 Swarovski binoculars and go with the bird, and then report them and then argue about what they saw. Turns out, though, that there are lots of divers that have been doing this for 30 years also. And this program, the Reef Environmental Education Foundation, has allowed divers to report their information into a centralized database. And part of my goal here is to try and figure out how to make use of those data in a way that can serve fisheries management. What these divers do, in a nutshell, is on the course of any given diving experience that they have, they will, over the course of their dive, write down every single species of fish that they can possibly identify, as well as relative abundance of those fish during the course of their dive. So single, few, many, or abundant. Single is 1, few is 2 to 10, many is 11 to 100, and abundant is more than 100. And then they report all of that. Used to be on ScanForbes back before the internet. Now they just go onto they use a specific application that you can install on your computer, or you can go and enter your data on the web. This is just a time series of reef data since its inception in 1993, just to give you a sense of where these reef surveys happen and their coverage in a global context. The reef program started in the Florida Keys in the Caribbean, and then that's why you see, oh my God, you cannot see that at all. It's too bright in here. Isn't Seattle supposed to be rainy? That's the Caribbean, if you can see this map. And then the rest of it is the rest of the world. So you're starting to see over time, leading up into 2025, the data expands most of the globe, at least in the tropical and subtropical regions. Not a lot of data from the Arctic or Antarctic yet, but who knows? There are hardy souls out there who do dry suit diving, so. This is what the Caribbean Basin looks like. So we've got a huge amount of data over the last 35 years from the Caribbean Basin. And it represents very powerful data set, especially for those types of questions you might be interested in asking and answering that are not for the commonly studied fishes that we generally think of in the Caribbean Basin, let's say bluefin tuna, some of the jack species, red snapper. Those are species that our federal government focuses a lot on when it comes to fisheries management. What about blue hamlets? What about conger eel? So there's a lot of species in here that divers are reporting on that we otherwise don't get a lot of good information. In places like the Florida Keys and southern Florida in general, we get a huge amount of data that's relatively consistent across space and across time for since 1993 onwards. And so I've been working with Reef and then working with the South Atlantic Fisheries Management Council to help support their efforts to tie that data into fisheries assessment work. Most notably, the CDAR process for the data workshop has been focusing on hogfish. Is anybody from this region of the world? No, okay. So if you were, you know a lot about the species. It's arguably one of the world's most ugly fish, but it's cool. This mouth opens up starting here. So it opens up like a giant levered trap, and they're invertebrates, but that fish is probably about that big. These are highly prized fishes in Florida and in the Caribbean Basin in general, and they're largely targeted with either hook and line or most especially spearfishing. So they are the focus of a lot of fisheries discontent because they are heavily harvested. They're not well assessed because there's not a lot of great information on them, because if you're not commercially harvesting them, you got to rely on the creel surveys, and getting creel surveys from some spear fishermen don't have things like parting boats, right? There's no such thing as a spearfishing parting boat. So you really have to rely on these creel surveys. Data is few and far between. So it's a challenge to assess this species. Nonetheless, it's not doing great. It's pretty heavily overfished. So they need information, data. So it's a good target for a survey like the reef data, because reef divers see these guys all the time on the reefs. So how do we help the South Atlantic Fisheries Management Council figure out ways to incorporate these data? In the tropics, and especially in the Florida Keys in the southwest, they use this thing, visual reef census. This is a federal, statistically designed federal fishery survey. It just happens to be with federal employees that go out and they do diving, and they write down every species of fish and the number of individuals. And they're not transects, they're these sort of cylinder-based approaches. It's kind of weird, but it's got a history. And this is the primary data they use for indices of abundance for species like hogfish. If you look at the locations where those surveys are collected, and you look at mean encounter rate, for instance, for this reef visual census technique, which is the federally designed survey, versus the mean encounter rate for reef divers, as a function of each one of these dots is a particular species. The details of the coloring are not really all that important. What you'll notice if you squint is there's a pretty strong positive correlation. So at a first blush, it suggests that they do a reasonably good job of capturing the same sort of dynamic in terms of relative abundance of individuals on a reef. Even though this reef survey is not a time survey, they write down metadata like how long they were diving and how deep they were and what the temperature was. So we have metadata for all of these dives, but we don't tell divers to do anything. We just say, go dive. And if you do, write down your things and then report them to us and give us your metadata. So that's the data we work with. With a postdoc of mine, now works with the DFO Canada, Dan Greenberg, we actually took both those two different sets of data, and we essentially did index standardizations for both survey types, and looked at the cross correlation in those things using effectively time series state-space models with random effects of things like surveyor, dive location, et cetera, et cetera, et cetera. So there's some fanciness under the hood, but not all that fancy. These are essentially just time series, and we're asking the question of whether or not these time series tend to track the same underlying process model. And by and large, they do. A vast majority of the species tend to favor a single state type model. So, great. Pretty good evidence to suggest then that we can rely on these citizen science data in order to do index standardization and generate indices for fisheries assessments. And so Dan and I put together a white paper for the CDAR working group, the data workshop working group, in order to create an index of abundance that they could start to use in their assessment. And the reason why this is valuable is because for hogfish in particular, the RBC data, the federal survey, goes back all the way to around the early 90s, like the reef data in the Florida Keys, but not in South Florida. So if you're off of like Miami-Dade, those data only go back to, whatever, about 2014. And so that, of course, is not a lot of time series in order to inform any sort of an assessment. And so one of the reasons why they were quite keen to use the reef data is with the reef data, we can walk much further back with, of course, a lot of uncertainty, but we can walk much further back in time. And by doing so, we can continue to leverage this RBC statistically designed federal survey back in time before it was built. I'll sidebar, I don't have a ton of time, but this row up here, this is a correlation between the two different time series and as an autoregressive process. Rather than just modeling this as a single state to which both observational time series inform that state, we're not doing that because they did not want that. They did not want the reef data to have equal to or more influence on the time series trajectory relative to the federally designed survey. They were really worried about that. So our solution to that was to say, okay, well, let's build a two-state time series model and allow those states to covary using this correlation coefficient. And that correlation coefficient ramps up or down depending on the extent to which these two time series agree. And to the extent that they very much agree, they essentially collapse the time series on top of each other. And this allows for them to only use the underlying state estimates from the RBC survey, but say that the reef data informs the estimates from the RBC survey. It's a weird modeling approach. It's a little kludgy, but it's driven by politics. They want to say that this time series is an RBC time series that's supported by the citizen science data. Right? So it's where the rubber meets the road in terms of where do you give and where do you take in order to get your data accepted for an assessment. And I don't know if a lot of you have done assessment stuff, but getting data into an assessment is an arduous process. There's a lot of work to be done to get there. All right. Criticisms. Well, great. Hogfish, very different things, right? I could see 100 of these things, or I could see one of those, and they matter very substantially. So just having the number of individuals counted doesn't give us everything we need to know. Of course, in a fisheries context, and you all know this, two key pieces of information we need from fisheries, if nothing else, how many fish are they and how big are they? But that size frequency matters a lot to us, and that citizen science currently doesn't get us there. We just get counts. So that is the motivation behind everything else that I'm going to talk about today, which is essentially starting with the SMILE project. And it is, we got funding from the South Atlantic Fisheries Management Council to develop citizen science approach to generating length frequency data as well as abundance for species of interest to the South Atlantic Fisheries Management Council, including hogfish. And two species that they care a lot about, black grouper and hogfish. Black grouper they care a lot about in terms of getting length frequency, because as it stands right now, any given year they may get 20 to 50 lengths from the commercial fishery. They have really low length frequency data to inform any assessment. And we've already talked about why hogfish is important. So all of these nerdy divers that are doing fish watching, they're not just diving, but they all go under the water with cameras, like literally all of them in a modern context. And it's because you're seeing, and you know burgers, they're just burgers and fish watchers of the same. There'll be a manta going over their head, and they've got their head stuck in a crack looking at a goby that big, right? Because that's the interesting thing. And if a lot of these people, like birders, skew older, and they need support for their visual acuity, and so cameras help for all of these reasons. You can take a picture of that tiny fish, blow it up on your computer and nerd out on it, or send it to iNaturalist or whatever to get a positive ID. So everybody has a camera. These are TG cameras. They're bulletproof because they're a waterproof camera inside a waterproof housing, and it's cheap. So this is pretty much what everybody uses. And so we start with, how can we leverage these things in order to get us size? In order to do that, we need to build a depth map, right? If we take an image, it doesn't tell us the size of something unless we can generate with that image a depth map from its field of view. And if we have a depth map, we can know how far a fish away it is, and then we can get at its size. So we developed this tool called the Smile camera. This is it in action. All it is that TG camera, but it's got a single laser mounted on the top of it. And as long as we do a bunch of calibration with that laser relative to the lens, that allows us to get depth of the laser at the point at which the laser dot hits something in your field of view. Just with the single laser. Okay. The way it works is it's a perspective thing. So if I have a pole coming out of my brain case, right? And I got a laser mounted on that pole. It's about 1 meter away from the center of my brain case. Right? Imagine the pole, laser. And I'm standing this close to the wall. In my field of view, that laser dot is going to hit way the hell out here, right by where I can barely see it. It's still in my field of view, but I can barely see it. As I walk away from the wall, that laser dot is going to come closer and closer and closer to my center of view, right? It's going to go to that vanishing point in the middle. And so if you kind of think about it in the context of in a three-dimensional image, the further away or closer to an image it is, you have that image kind of walks across your frame of reference. If you think about your frame of reference as a number of pixels wide by a number of pixels deep. And so we can, as long as we know exactly where that laser is mounted on that pole and its angle relative to some known depth, then we can, by calibration, we can calculate exactly where that thing is. And then from everything else that we hit a laser dot from then at that point on, as long as it remains calibrated, we can estimate the depth of that object that is hitting. Once we get that, we can generate lengths through image processing, through something like ImageJ, which probably some of you have used. It's a pain in the butt, but we're going to talk about automating that, but we can do that. And so this is just some preliminary data with our Smile camera for areas of hogfish in protected sites versus non-protected sites, and then comparing that to the data from the RBC, because while the way that they do lengths in the federal survey is they go down with a T-bar and they just estimate right down the size of those fish, which I would argue is a terrible way to do length estimation, but they swear up and down that it works because they do validation and testing. Anyway, if you look at these two things by and large, they match. This is really important because even if we are not measuring all fish everywhere to get a full length frequency of the entire population, which would of course be the thing we absolutely want from a fisheries assessment standpoint, at least knowing that the length frequency of the fish that reef surveyors are seeing and RBC surveyors are seeing matches tells us that effectively the selectivity of the two surveys is about the same. And it gives us confidence that we can put the two time series of abundance together into the survey. Into the time series or index of abundance. So it's a valuable just on its face. Problem. Calibration is a pain. You really got to calibrate these things every dive in order to get them accurate. And we realized quickly that citizen science divers have no interest in spending the first 10 minutes of their dive shooting a calibration slate at various distances away from it underwater. They don't want to do that. And then even after they do it, somebody's got to sit in the lab and process those calibration images to calibrate the camera. It's a pain in the butt. So what's our solution? More lasers. One's good, two's got to be better. It must be true. And so we developed this new device here, which is a dual laser system. We actually did this with a collaboration with Backscatter, which is this sort of the elephant in the room for underwater cameras and lights. And they have a production company over in Asia that makes all of their equipment. We work with them to produce this underwater dual laser system that is ostensibly exactly 2.5 centimeters apart, the two lasers in this thing. You can't see it because the light sucks, but there's a dot and there's a dot, those are the lasers. Bulletproof, it's got external charging, single button on off, works great. Holds calibration so that their parallelness doesn't really change if you bang them around a lot. It's really, really hard, it turns out, to get two lasers perfectly parallel in a mounting context. It's not a problem to get it like 2.5 centimeters apart, a meter away from a wall, but once you move to this far away, those things remaining 2.5 centimeters apart when they hit the wall, it's hard to calibrate that. And so while this production company is really good, they're not that good. But it turns out through a little bit of math and trickery and using what we know to be true about the pixel dimensions of an image, we can account for slight imperfections and parallelism through image processing. Importantly, without having our divers have to do any calibration at all, except for calibrating the unit once. You can even do it on land. Okay, cool. So with those new laser calipers, now as long as we just get two dots on a thing, we can now measure the size. I should step back and say, laser calibration is nothing new, right? That we've been doing that forever. If you see any submersible dive on YouTube where they're looking at some Dumbo octopus or whatever, you always see the two laser dots moving with the ROV, laser caliper. So they're everywhere. And the reason it works is because as long as they're perfectly parallel, those two dots are always exactly the same distance apart, no matter how far away or close to an object you are. And as long as you know the distance between those two dots, you can estimate the size of whatever it is those two dots are. Simple, right? Okay, so we're all about making this easy because you got to make it easy for citizen scientists. So making it easy means we got to get lengths from the video. And I should point out that we're going to this system here away from this TG. This is just GoPro. Cheaper, smaller. And now instead of taking pictures, we just say go when you start the dive and stop when you're done with the dive. That's all you do. And then you just go around getting lasers on fish, which turns out is not easy. Because fish either really like lasers or they really don't. They think it's food or they think it's going to eat them. One of those two things is true. So, but you gamify it a little bit, right? And it works okay. So how do we take a crazy amount of data, image data from these things? Because they're, 30 frames per second. If you're at low resolution, 30 frames a second, how do you take all of those frames from a 1 hour dive and turn that into measurements of a few fish over that time frame? And so that's where AI and image processing comes in. So we've been over time, we've been slowly chipping away at our AI workflow in order to tackle this problem. So just kind of briefly walk you through it. I'm going slow. You start with raw images or a video frame. Probably can't even read it back there. Sorry. You start and you input your raw video data, and you split it up into a whole bunch of images, frames. Let's say one every 5 every second, something like that. So we downsample it a little bit, and then we throw it through a first order detector. This is just a boxing algorithm, like a YOLO algorithm. So it just says, okay, we train that algorithm to find fish, and then we run that algorithm on all of our frames, and it goes in and it boxes all the fish that are in there. And then from there, we can pull out some of that tracking data. We're going to use that to save later. We then move forward on these detected bounding boxes. We can score and track them, including doing species classification. Scoring and tracking them, and we'll talk a little bit about this. This now gets into what Marcus is going to be working on in this world. But scoring them is, some images are of a fish kind of doing this. Well, you don't want to measure the length of a fish when it's doing that, right? You want it to be perfectly like side on, nice, straight, flat, like it's on the length board. That's what you want. And so if you've got 700 images across 30 frame per second video of the same fish, you want to hunt for the best or the few best of those images for length estimation. So we've got to score all of those 500 images that we've now boxed for the quality of their fit for purposeness when it comes to length estimation. So that's what our scoring algorithm is going to do. It requires us to generate a whole bunch of training data set where we score images of fish based on how perfectly parallel and arranged they are in the camera. That's where a lot of undergrads come in, or 199s. Nowadays, they have to get some version of class credit for it, or you pay them, one of the two. So, but we do that, we get a whole bunch of training data set, training for that. And then we also track individuals across frames. And this becomes important because we need to be able to track an individual from one frame to the next so that we know it's all the same fish. And that tracking algorithm was something that we're working on now. We'll talk a little bit more about that later. And once we scored and track these boxes, we do a segmentation on them. Segmentation just means, okay, we take the YOLO boxed fish and we do a mask on the fish, right? So if you run your iPhone, you click on something on an image, you hold it long enough and it kind of flashes and then you can move your mom out of a picture, right? So that's what we're doing, but with fish. We're creating a mask to outline a fish through the segmentation. And then once we've done that, we can extract endpoints of nose and tail. And then we also need to detect those laser dots. So we can estimate once we've detected them in image, we can estimate the distance between those two laser dots. And with the head and tail and the known pixel distance between those two laser dots and known actual distance of the laser dots, we can generate length estimation of each individual across frames because we tracked it. We can integrate back into this optimal tracking. And then we got this final output where we've got by species, we've got a whole bunch of images of single individual tracked fish that each of them have measurements, right? And this is being organized by an AI machine learning person that we're working with, Sunny. So think about it in terms of this is how it works. So we got some random image that we pulled from a frame, and we've now done the YOLO boxing on these guys. And you can see this ID 36, this is just one frame of tracked fish number 38. So if you walk back or forward in time in terms of frames, this fish moves across in the frame. And then the score here just is like, it's the arbitrary rating score that the AI uses to say, is it a fish or not? So 0.76, yes, this is a fish. This is 0.11 fish, less likely to be a fish. So you have to decide on what your threshold is. Beyond that, what we've done is we say, okay, well, ignore all fish except for those that are plausibly in the range of where our lasers are. Now, if it's not in the range of our lasers, ignore it. This cuts down on our AI pipeline computational intensity that needs to happen. Once we've done that, we say, okay, it's boxed, it's in our pipeline. Now we do masking or segmentation. This is just a couple of different segmentation tools. It turns out segmentation is hard. Like any other AI, you need training data set, and imagine telling an undergrad to generate this segmentation for 5,000 fish, right? It's a pain in the butt. But it turns out there are several different segmentation tools out there. Google segment anything. Apple has a segment anything type of implementation. This is why you can move your grandma out of a picture, right? There is a company called Fishio that is focused on like trophy fish stuff and for fishing, for recreational fishing, and they've developed a very robust AI segmentation for fish, and that's what we're using, and it works pretty well most of the time. So this is an output of that Fishio segmentation. Then we use some sort of pretty standard geometry to get at sort of the axis of the fish in order to estimate the length in the head and the tail. And this then allows us to get at the where it crosses the segmentation mask. That's where we get to our lengths of fish. Okay, and then finally, in order to turn that length in head and tail into the total length of a fish, we have to identify using AI where the dots are, laser dots are, so we can pinpoint the center of those laser dots, measure the distance between the two in pixels, and then allows us to translate all of our pixels into total centimeters. And at the back end of all that, and you can't see it back there because of the light, but this is an output. So here is individual fish number 18. It's got across 30 different frames of this individual fish. It's got an average or a median centimeter estimate of 57.91 centimeters. And this current frame image of this fish is 60.92. So you get some estimate of length plus uncertainty across all of your measurements. Okay, cool.
I have a question.
Yes, please.
Is this, so like these pictures you're showing, I don't know, do you know how deep? I was thinking like based on kind of the color, like I know colors change a lot depending on how deep in the water you are.
Yeah.
Is this like work only at certain kind of depths that you're at, or is it pretty good at like finding those outlines even when it's just kind of black and white?
Yeah, great question. It's only as good as the training data set we give it to do the detection part of it, the boxing part of it. So we have done, this is all data from our grouper project down in the Caribbean. Again, this is what Marcus is going to be working on. We actually developed a huge training data set for these types of images to make sure we were doing it okay. It turns out that there are all kinds of people doing fish detectors in the world, and nobody has done a good job of organizing them into sort of a centralized way. Everybody kind of, even NOAA, it's very proprietary. You would think that because they've got a lot of training data sets for AI. NOAA's leaning heavily into AI. I don't know if you've ever used Viame, but like they're good luck getting their training data sets. I don't know why they're not sharing them, but they're not. And so it's really hard to get more generalized training data that would make it do better across a diversity of environs. Yeah, it's a good point. Yeah. Somebody, I think, needs to take the bull by the horns and make the entire community sort of create a generalized shareable database. This is just to show you proof that it exists as a proof of life of a GUI application that we've been developing that allows people to step through a single video that we push through the GUI that runs the whole entire pipeline and allows us to do an approval process. So right now there's still some human intervention involved. We're trying to move towards push a video and then out pops a list of fish by species and their sizes. That's where we're trying to get to. But we're getting closer. Again, Marcus is going to have to put it over the finish line. Sorry, Marcus. Let's get to it, would you? We haven't graduated. Okay, so that's the general SMILE project, and but we're trying to apply this to. This would be way cooler if it was dark in here, but this is a video of an aggregation of this species, Nassau grouper. It's an endangered reef fish found in the Caribbean. It's endangered because it does this. It forms these really large reproductive spawning aggregations that are really predictable in time and space. So if you can find them, you can fish the crap out of these guys with just a couple of boats and hook and line until they're all gone in a couple of years. And so that's happened throughout most of the Caribbean Basin, and there's only a few locations where there's large aggregations for these things remaining. And the Caymans is one of those places that still has large aggregations, and there's a whole reason why that is, but I won't go into it now except to say that the Cayman Islands government was pretty proactive at recognizing the problem and fixing it before it became an extirpation event. So we've been working with the Cayman Islands government in order to study this population over the last 20 plus years in lots of different ways. Marcus is going to work with this. I'll keep talking about you, Marcus. No expectations, of course. He's also going to do to help out Rick Nemeth and the folks in the U.S. Virgin Islands to do population estimation from their population as well, which turns out is recovered also. Population estimation is hard. If you were looking at that video, let's just look back at it now and you can think about it. How many fish are in this video, right? Like if you're within 1,000, 2,000 fish, I would give you a cookie because you won't be. It's really hard to do counts of fish in this sort of a situation. So the way that we do it historically has been through marker capture. Again, you can't really see it, but there's a flight tag of the fish. Send divers down, tag, let's say 100 fish in the population, and then over and over again, divers count them 50 fish, and they keep track of how many of those 50 fish have a tag over and over and over again. And if on average across all of those things, 10% of the fish on any given count on average are tagged, and we tagged 100 individuals, well, it must be that 100 individuals is 10% of the whole population. Population's 1,000, right? So that's the Lincoln-Peterson estimator. We just invented it just now. So that's how we've been doing it, which works great. But all we can tell here when a diver is looking at a fish is it tagged or not? We can't say that that's Bob, and we've seen Bob before, and Bob grew 3 inches. Bob likes to hang out with Nancy. Bob never comes in January, only February. We don't have any individual information about fish, and boy, would that be handy to get at everything from skip spawning to growth rates to individual behavioral preferences, et cetera, et cetera, et cetera. So powerful if we can unlock it, but we can't with this tagging approach. Here's an example of it. Again, I'm not even going to talk about it because you can't see it. So problems. Tagging is not inconsequential. Sometimes we're tagging them with pole spears underwater, and we're pretty good at it, but nobody's that good. Sometimes you miss, you hit an air bladder or whatever, and it's not pretty when you do. You ever see a fish get hit with an air bladder underwater? It doesn't look good. It just looks like you just popped a tire. So that's not good. And of course, there's also there's evidence for some of these Floy tags getting infected. If Floy ever tells you that it's non-impactful, they're lying, because we go and we see these fish 3 years from now. Some of them are not looking great. Mostly the tags fall out. Again, Floy will tell you they never do. Probably 80% fall out within a year. Some of them stick around a long time, but that's a long tail. They lose them pretty quick. So that's another thing, right? These tags are not long in nature, and also they're ugly, and divers hate looking at them. You imagine, this is the reason why Cayman Islands wants to keep these group around. You see a fish that big on a reef, this big wild natural tiger of a fish. People pay big money to go to have that experience, and if it happens to have jewelry, you don't like it. They don't like it. So we need to try and avoid that. Well, it turns out, here's a nice, big, beautiful, well-identified fish. These patterns are both unique to an individual and they last. They're permanent, from this big to that big. This is handy because we can use facial recognition, essentially, keep track of an individual as long as we can get good quality images of individuals from a video. That's what AI thinks fish facial recognition is. So it turns out that we've been collaborating with a couple of companies, Wild Meat or nonprofits, Wild Meat and Conservation X Labs, that are really kind of leading the charge on this individual instance pattern recognition. They started on things like zebra and cheetahs and things that people care a lot about, not thinking a lot about fishes. Of course, we think a lot about fishes, and it works pretty well. So we've been doing that. I'm just going to give you some examples, just hopefully to convince you that it works. So here's a fish that's 1 month apart. AI or this algorithm says this is the same fish. So if you see this kind of pattern here of like 4 corners, you see it here too, right? So if you stare at this thing for a while, I believe that you will convince yourself thoroughly that this is the same fish 1 month apart. And that's what AI says is the high regions of match. Here is a fish that is 7 years apart. Back in 2012 versus 2019. First thing you should note is, man, our cameras have got a lot better over time. But if you start to stare at this thing again, not great because of the thing, but you can, there's areas here, this like weird Italy thing here, you can see the weird Italy thing here, dot, triple dot here, triple dot, it's the same fish, 7 years apart. So we've got pretty good evidence now that we can not only track individuals across long windows of time, but we can capture a lot of information by just simply taking high quality video of images on that aggregation site. Because we care about citizen science, we created this as a citizen science program that supports our science-based approach. So any diver anywhere in the Caribbean Basin can now participate in grouperspotter.org. They can take a picture of Nassau grouper, submit it, and it goes into our centralized database. It will also be part of the database that Marcus is using with the Virgin Islands, et cetera, et cetera, et cetera. So, and no, they don't move islands. So we've got 20, this is me. God, I was handsome, look at me. So that's 20 years ago. Mark, maybe this is when Mark and I were in grad school. Remember that, Mark? We with brain dogs. This is Brice with brain dogs body form. He's just used to me on an alternate for back when Mark was in grad school in 1942, that was the state of the art, highest top level prosumer grade camera. That thing was bad ads. It used digital videotapes. That was hot stuff. Hot stuff. You'd have to buy those tapes and you put them in the camera. You young people don't understand what videotapes are, but they were a thing. And if you got a good one, it was 120 minutes long. This one's only 60 minutes. This one's cheap. And so you get your video camera and you take it. And this was our laser caliber system back then. So we had this aluminum machine thing and a couple of laser pointers. Okay, so from then to now, we got a huge amount of terabytes and terabytes of video data. And those data have been used with things like laser calipers and now in a modern context, stereo video with GoPros, allow us to get at length frequency distributions of that aggregation through time. And of course, you all are fishery scientists, you know length frequency is helpful, not just for how many fish there are, but how much biomass there is, but also a key thing in a stock assessment context is it allows you to estimate recruitment, right? I mean, recruitment is largely informed by length frequencies. So it's been very helpful for us to inform the Cayman Islands government to say things like, it's not surprising you haven't seen a lot of population growth in the last couple of years. Recruitment is very episodic. So it's important pieces of information, but it's really hard for us to get at because we have to have people stare at video screens to do it. So we're going to try and use AI to fast track a bunch of this. Our challenge here is that, you can't see it. But this is boxing. This is one undergraduate. Actually, I think this was me because I actually do something with students. Every time I graduate students, I do this rather than read their papers. So that's all of the boxes that are hand drawn on one frame. And this is part of our training data set, right? So we do this on thousands and thousands of frames, and that's what informs our YOLO detector for our boxing algorithm. It turns out that the more individuals in a frame there are, the lower the quality of, or the lower the scoring is for the detector. It says, if I have one fish among 400 other individuals, it could be these, as your eyes look at it, the exact same quality of image for boxing, but it will give it a much lower ranking score within the detector algorithm. We don't understand why, and it's one of those things we're trying to figure out. But it creates a challenge because we can't give it a single threshold. We can't just say anything over 0.7 in the arbitrary scoring of detector, keep, otherwise throw away, because a 0.7 when it's on its own would actually be a 0.1 in a school. So we have to figure out how to deal with this problem. And it's not clear. There's a lot of things with AI, right? It has its own mind and you don't even know why it's doing what it's doing. It's weird. It's like we're studying behavior of a zebra, but it's AI. It's crazy. All right, the other thing is, here's a still frame from one image of the most modern video that we have. And this is just to say, if you just zoom into this one area right here, just that one little quadrant of this single frame of a many tens of thousands of frame 5-minute video, we have, if you were to be able to see this, at least 20 usable images. So the volume of pattern matchable, recognizable individuals in these videos is truly massive. And it requires that we train our AI in a way that accounts for the very high resolution of the modern imagery that we're getting. All right, that's a challenge. This, like we talked about, the YOLO training, the fish underwater training set is hard. So I'm not going to talk more about that, but it's not easy. Tracking individuals is also a challenge. So here's an individual that AI thinks is individual 14. At some point, it loses track of individual 14, and now it starts calling individual 15. It's the same fish, but we lost track of it for a second. So our tracking algorithms aren't the best. Is that a deal breaker? No, but it means that at least some of the time, some of our individuals are going to be the same fish. And so can we improve that? A lot of people, if you're using Viame, for instance, I don't know, does anybody use Viame or know what it is? Okay. All right, so if you're using Viame, you can do a tracker, you can't train the tracker. Get what you get, you don't throw fit. It is it. So there are, we can, and we are doing this now, we're building and training our own trackers, but it's it's an intensive process. Yeah.
But like that problem you just had there, presumably you can use some autocorrelated aspect of that. I mean, if somebody was here, last frame, and there's a fish here this frame, I mean, did this one literally disappear and another one appeared there de novo? Or is your algorithm good enough to understand that within some margin of error, they have to do the same thing, because.
The way that it uses it is it tracks an individual across frames by looking at sort of trajectories. And the problem is if that trajectory breaks in a meaningful way, if it goes behind another fish in an aggregation, for instance, or it goes behind a rock and then comes back out again, the algorithm at present is not smart enough to go, okay, go, disappear, go. It doesn't like that disappearing part. So I'm not sure how to fix it. I don't even know if it is fixable, but it's a problem.
My next question is, are any of these fish social with respect to, like, do they hang around with friends, cousins, brothers, sisters? Right from your ID perspective, is there some group membership that helps inform that ever, or is it always we draw from the entire deck of cards?
Yeah, me, that's what Marcus is gonna figure out for us.
Yeah, I'm excited.
He's got a lot to do. I don't know what he's doing sitting here, to be honest. It's just, he's got work to do. So, yeah, I think that's an interesting question. And I think that's something that we, it's pervasive in sort of social animal culture, but it's not been well demonstrated ever in fish as far as I'm aware. So I think that, to me, that's a really interesting question. And I would bet you a large amount of money that there are social groups that are active out here. Yeah, it'll be interesting to see that. All right, and then this is just to say that, so our problem here is unique but related to our other problem, which is in the one case we already talked about, we're trying to identify the best image where the fish is perfect side on and measurable, like it's sitting on a length board. That's what we wanted when we're getting at lengths. In this case, our scoring criteria where we're scoring, this is the same fish across multiple frames, and each one of these has a raw score based on a training data set we built, where the training data set now is specific to how good is this image for facial recognition. So now we're scoring images not only based on their measurability on a length board, but also how fit for purpose any particular frame of a fish is when it comes to feeding it into our pattern recognition tools to make sure that we are capturing that individual accurately. So this is, again, something that you're gonna, we talked a lot about, this is further along than when we last talked, which in some senses is good, right? But we're gonna have to get it over the finish line. Don't worry, Marcus, we're doing it. It'll be fine. Okay, and then we don't just do NASA grouper, right? So for the grouper spotter, we've actually expanded out to Goliath grouper. This is a really important species for the tropical Western Atlantic, also critically endangered. These are the guys that get basically as big as a VW. They get really big, 800 pounds plus. They get huge. And they form aggregations that are really easy to fish out. So they've been recovering slowly. They've been protected since the late 70s, early 80s, completely in Florida. And they've been recovering slowly. Over time, when really large creatures that love to eat other creatures are gone for a long time, and then they start to recover, be they sea lions or Goliath grouper, there is conflict. Because anglers don't like it when the fish that they're targeting get depredated, right? And it's a problem we're going to have to deal with over and over again in a conservation context, where conservation meets fisheries. This is an example of that. Oh, yeah.
So wait, sorry, what does depredated mean?
It means you got a fish in a line, you're like, yes, I got my red snapper, this is gonna be up, damn it. And then big Goliath group raid. That's depredation. Nobody likes it. And sea lions do it all the time in everywhere, but especially in Southern California. So there's constantly sea lions washing up with like shotgun holes in their head.
Wow.
How did that happen? I don't know. So it's a problem. So Florida has recently opened a ticketed season for these guys. So like bears in Alaska, you enter a lottery, and if your name gets drawn, you get a ticket to harvest one of these guys. And it has to be within a certain criteria. It has to be a certain size, it's a slot. Can't be the babies, and it has to be in a certain area, but it's a ratchet effect, right? So, you start opening it up and opening it up and opening it up. They did that mainly because there are issues, conflict between anglers and the fish. But at the same time, we don't know a lot about, from a fisheries context, from federal data, we don't know a lot about the species. Why? Because it hasn't been legal to catch them since the 80s. So there's zero fisheries data for these guys. So observational data, like from reef, is the best we got because there's nothing else. Divers just looking at them underwater, it turns out it's pretty powerful data. Wouldn't it be even better if we could start to do marker capture on these guys and get population estimates? And we can, using things like the grouper spotter program. So we're doing that. And then all the way over the Pacific Islands, you probably have seen the National Geographic with these guys. These are camouflage grouper. You probably saw the video of like white tips. Like white tips are like the hyenas of the Pacific, like mobbing these guys and eating them. It's graphic and horrific, and it makes for great TV. So you should watch it. Anyway, we're starting to do it with Goliath Grouper as well through a private foundation that's interested in aggregation on Fakarava, which is in French Polynesia. Amazing place if anybody gets a chance to go. I always tell people, sidebar, go to the Pacific and see a reef now, because you're not going to see it in 10 years. The Caribbean is done for. Don't record that. You can delete that out later. It really is. I grew up and everybody's like, oh man, the rainforest. And yes, the rainforests are going away. The Caribbean is gone. It is literally not a reef system anymore. You're welcome. Little nice Friday gift. The Pacific still has some places that are very high quality cover that still look like what a reef should look like. Fakarava is one of those places. Go, spend the money, get there soon. And this is not AI. This is an actual image, still framed from a video from the aggregation of these guys. You can imagine the challenge here that AI is going to have. Right? They are built for camouflage. They're evolutionarily adapted to avoid AI detection. So, and shark detection, which is basically AI with teeth. And that's our future, by the way. So, yeah, so this is going to be a feature challenge. Hey, Marcus, you're up. All right, that's what I got. Thanks, everybody. I appreciate it. If there's any questions, I'd be happy to take them. Otherwise, go and eat or whatever you need. Thank you. Please.
Thank you. This was super cool. I was curious and going back to the beginning of your talk, thinking about these methods to build indices for stock assessments, right? How do you handle sort of like the catchability, if you will, of your index, right? Because presumably I would think that divers would, go to places where there's bigger fish maybe, or just more fish, because that's what is fun to look at. So how do you incorporate, I mean, obviously it is preferential sampling, which you guys handle, I imagine, but kind of like, I'm thinking what's Q in your index?
Yeah, so, a good question for those of you online. The question is, how do we handle catchability in Q when it comes to our index that we generate from citizen science diver-based surveys? I'll go back to kind of what I had said before, which is in some sense we're punting a little bit. Well, first of all, we're doing an index standardization. So we're using the exact same machinery that one would use to take recreational catch data or commercial catch data and account for the fact that effort is uneven and we can't tell people to go survey particular areas. We get what we get data-wise. And so from those data, we have to do some version of standardization to account for things like uneven effort as a function of habitat or location in order to back out some reasonably unbiased estimate of abundance. And so we do the exact same thing with divers because in a sense, it's the same type of data. We're not telling divers where to go. They tend to want to go where it's fishy, just like anglers do, right? So we use the same techniques that we would for standard index standardization approaches. Beyond that, the catchability problem, we punt on a little bit. And what we say here for the hogfish, I would argue, and I did argue, that our bar here is not, can we determine catchability? Our bar is is it the case that whatever the catchability is, it's essentially the same for the federally designed survey compared to what divers are seeing? And I'm arguing that because our length frequency that we're capturing is approximately equivalent to the length frequency of the surveys that are done by the feds, that our catchability is approximately the same as whatever theirs is. And by making that argument, we can then say, okay, well, if we're seeing the same length frequency of animals in any given year between the two surveys, it's probably a reasonable assumption to make that whatever the catchability, it's equivalent between the two survey methods. Therefore, we can put them together in order to improve our estimate of that population index of abundance across time. And then catchability becomes a problem for the assessment folks. They got to estimate it somehow. Does that answer your question?
Yeah, so you make one index in the end of the federal survey and the citizen science survey.
Yeah.
Okay.
And especially because of the way we did it, where we actually are mapping two different state processes that are allowed to covary, where we estimate that covariance term. They only use the index of abundance from the federal survey. It's just that index of abundance is informed by that covariance term across the other state process. So it gets us out of the business of saying, use the reef data. All we're saying is, hey, the reef data more or less inform your estimates. You should probably leverage them.
I'm not seeing any questions online.
So I'm curious about it seems like you're doing a lot of collaboration with some conservation groups, and presumably from a lot of people I've met who are divers and like very concerned with conservation. Has there been any work on or any consultation on data that they are collecting or the funders are giving money to be collected will be used in a stock assessment that could then lead to opening up fisheries? Like, do they know that the submitting stuff for this is not just from a conservation? It's like if we do this index and it says, okay, well actually these fish are doing better than we thought, so now you can catch 500 Goliath grouper next year.
Right. Well, I would assert that you're supposing that opening a fishery is not a conservation action, and I would argue that it may well be. So if we can, with best available data, if we can assert that a fishery is recovered to the extent that it can support a fishery and still be healthy and still maintain itself into the future and maybe even still participate in an ecological context within the ecosystem itself, that to me is conservation. We're informing wise use of a resource while ensuring that it is there into the future.
But do they see it that way? Be collecting the data. I agree with you completely.
Yeah.
If it can be fished, it should.
It's an interesting question. I will say that from Brice's perspective, they are a non-advocacy group. They're from the jump. They have always been. The program has always been. We generate data that's useful for science, not necessarily that we take a stance on a particular conservation issue and we lobby for it or anything like that. So I don't know if it would come out of left field. I'll say that in general, anytime that we say that their data are being used for management, people get super happy because that's what people really want. It's not just that they want to generate data, and part of it is like they're listers, right? Like they want to see that, I don't know, wide-banded goby or whatever. Like they care about that, but they also really care that the work that they're putting in is being used in a way that is supporting better management, wise management. So all of my interactions with people when I tell them stuff like this has always been very, very positive. Okay. Yeah. Thank you. Mm-hmm.
So we do have an online question. Ray Hilborn asks, is there evidence that fish in areas with little human activity are more visible to diver surveys, particularly where fishing pressure is high? The fish tend to hide and be less mobile.
Yes, there is. So we go back to this catchability thing, and we're opening the hood a little bit up on it, right? And I do think that that could be a problem. What Ray just said. Here's another one. Everybody heard of the Gardens of the Queen, Jardines de la Reina? No. Okay. South side of Cuba. It is a protected area. It is like Mecca for divers. It is the place that everybody says like, it is what the Caribbean was 100 years ago. Is there's just sharks freaking everywhere. Gosh darn anywhere. And giant grouper and what, and it's true. If you go there, if you make the effort to get there, and it's not easy, there are sharks and grouper everywhere and really big ones. To Ray's point though, why? Because a Caribbean reef shark naturally has no interest in being near a diver. Like there will be reef sharks everywhere around you as a diver, but you'll never see them. They're really good at not being seen as a general rule, but not there. I think it's because of it, that there's a lot of feeding that's happening. There's a lot of feeding that's happening inadvertently or intentionally. Inadvertently, if you're on a night dive and you're shining a fish, your light on a dog snapper, and a Goliath grouper is shadowing you, as they do, it's a free meal. You blinded that fish, it's gonna take it out. So it behooves them to be really close to you. And so that's an example of where some dive places, there are attraction or repulsion to divers based on what's happening in the water. Whether or not that's spearfishing, like spearfishing is a great example. You go try and find a Nassau grouper in the Virgin Islands, there's not a lot of them, but I tell you, the ones that are there, you'll never see them. They're really good at not being anywhere near a diver, because if they've lasted that long, if they've gotten that big, it's because they've been able to avoid spearfishing. So that's an example of yeah, fish aren't dumb. You know, everybody thinks that fish is just these stupid things that fly. They're really actually quite smart, a lot of them. Okay, that was a long-winded answer to the question. Sorry, Ray.
All right, well, thank you all so much for coming. That was a wonderful presentation. Thank you, Dr.
Absolutely. Thank you all.